Okay, welcome back. This is part 3 of our virtual session. So in part 1, we looked at the microbes that are coming in on the grapes from the vineyard that may or may not be causing spoilage. And then we talked about SO2 and how SO2 works on those microbes or doesn't in order to help to prevent spoilage. In our second video, we looked at the use of chitosan and how that could be a good partner to SO2 in knocking back some of the population of spoilage organisms that we might see. And then in this part 3, we're going to be looking at using non-Saccharomyces yeast in order to either partner with SO2 or use in addition or in place of SO2 in order to manage that microbial population. Now you may be thinking, wait a minute, if I non-Saccharomyces yeast, didn't you tell me in video number 1 that non-Saccharomyces yeast were the exact creatures that were causing the spoilage to begin with? And you have a good memory, that is true. The answer to that question really is that it depends on the non-Saccharomyces yeast. So as we looked at in video number 1, there's lots of different organisms that are coming in on the grapes, and some of them will cause spoilage and some will not. And some will not in certain circumstances and will in other circumstances. And so when we think about sort of what set of decisions we're making when those grapes come in, what we're really trying to do is manage the microbial population. We want to encourage the good guys and we want to inhibit the bad guys. And that's really what the use of non-Saccharomyces yeast for bioprotection is all about. So the use of non-Saccharomyces yeast for bioprotection is not new. This is something that we've seen in use for many years now. If you look at your favorite catalog from your favorite enological supplier, you will probably find that there's 1 or 2 or 3 different non-Saccharomyces yeast products available for somewhat different purposes. And so that's the one thing, if you want to use this for a particular purpose, again, much like chitosan, I highly recommend talking to your representative to make sure that you're using the most recommended product for what you want to accomplish and to make sure that you're following the directions in terms of the timing of inoculations, both for your non-Sacc yeast and for the fermentative yeast that you need to use after that. And it's an important thing to highlight here that when we're talking about non-Saccharomyces yeast, we'll see in just a minute, these are not meant to be doing the primary fermentation of your juice or your must. This is something that's meant for bioprotection early on or for other purposes early on, but you still will need to use a Saccharomyces yeast in order to complete fermentation. So when we think about non-Saccharomyces yeast for bioprotection, what are the things that we need to be thinking about? And the first question is when should I actually be using this? Is this something I need to use all the time? And basically the answer to that is that these could be helpful at any time that there's a delay between harvest and the beginning of alcoholic fermentation. So if we think back to that graph that we were looking at earlier, we sort of have this delay between the time that the grapes come off the vine to the time that Saccharomyces takes over and produces that anoxic environment with the low pH and everything else that's going to be inhibiting our spoilage organisms. So that can include transport, particularly if you're picking your grapes in one region and trucking them across the state to another region. Those grapes might need some protection during that time. Settling. So when you're cold settling, we make it cold on purpose to inhibit any of our spoilage organisms. But if you feel like you've got kind of a big microbial load coming in, this could be an additional tool for that. If you're doing stabulation, you're elongating the time between when you're actually pressing those grapes and when you're starting your fermentation, and non-Sacc yeast might be helpful there. Cold soak is the most common use of this. So again, once we've sort of cracked open the grapes and we have it sitting there as must, we're intentionally elongating that period of time in order to try to extract additional color and additional aromas from the skins there. We don't want fermentation to happen. That's a big danger zone for spoilage organisms. And then I put in here an uninoculated fermentation as well, at least in my experience with these fermentations. A lot of times they do take a little bit longer to kick off. And part of the intention there is to give the rest of the microbial community some room to work. But again, you have to be careful there that you're not dealing with some spoilage organisms as well. So depending on why you're doing your uninoculated fermentation, you may want to consider using some non-Sacc yeast as well. So what makes a good bioprotection yeast? So I said before that some of these non-Saccharomyces yeast are spoilage organisms and some of them are not. So how do we kind of think about selecting for the ones that are going to have the best bioprotective capacity? So the first thing to think about is that we do want to select for non-Sacc yeasts that have a good tolerance to SO2. So again, you maybe end up using these in the instead of using SO2, but they can also be used in addition to SO2. So both the scenario of 2018 when we had all those microbes coming in and 2019 when we had significantly less microbes coming in, um, basically it's up to your judgment as to whether you want to use SO2 or not. But again, thinking about this period of time where we've got a lag between harvest and alcoholic fermentation, the idea is we want to knock back the other population with our SO2, but we don't want to knock back our Saccharomyces. Well, if we're using SO2, we don't want to knock back our bioprotective yeast either. Good implantation, and that's just a different language for saying if I'm going to inoculate with this, I basically want it to reproduce quickly so that it can outcompete the other native flora. So remember, we talked in video number 1 about how this is really kind of a numbers game, right? If we have a higher number of microbes coming in, then our Saccharomyces is sort of fighting against that higher number. And if we have any lag time in there, we're basically giving that higher number of microbes longer to be able to take over some of that ecological space that we've just created by crushing our grapes. So the idea here is that if I inoculate with a non-Saccharomyces yeast, that it's quickly going to reproduce and essentially hold that ecological space so that the native flora doesn't really have any space to be able to reproduce and take over. These also can sometimes inhibit the native flora by producing toxins, but again, we want to look for ones that inhibit toxins against spoilage organisms and do not produce toxins that inhibit Saccharomyces, so we have to be careful about those toxins. We also want them not to be fermenting. So for whatever reason, we don't want fermentation to be happening here. So cold settling, we do not want fermentation to kick off, stabulation, cold soak. These are all times where we don't want fermentation to start. So we're looking for a non-Sacc yeast that's not going to start fermenting in whatever environment we put it in. And again, that's going to allow us to get that time that we're looking for whatever reason. And it's also going to allow us to select the yeast that we want to be actually completing our fermentation in the longer term. And then it's important to say that we need something that doesn't deplete the nutrients. It's been seen many, many times in ambient fermentations that sort of lag phase where the native flora are working. They might be doing all kinds of good things, but they're oftentimes also depleting the nutrients because their metabolisms continue to work on this rich resource of grape must and grape juice that they're sitting on. So when we're looking at our non-Saccharomyces yeast, we want to make sure that it's not eating up all the nutrients that our yeast are going to need later in order to complete fermentation. So there was, I think this is well looked at when Dr. Osborne came to talk to us at the Virginia Wineries Association technical meeting I think it was in November. His lab did some work looking, just basically testing different commercially available non-Saccharomyces yeast and their ability to interact or inhibit the work of Hanseospora uverum, which remember that's the same as Cloacra apiculata. So what they did is they sort of did a monoculture of Hanseospora. They looked at the number of cells that would come up with that, and then they co-cultured it. So put it together with many different versions, and you can see we've got 3 Metschnikowia strains, 3 of this thermotolerant strain, and 4 Torylospora strains. So they co-cultured those to see, does this non-Saccharomyces yeast actually have an impact on the growth and acetic acid production that we would see from Hanseospora. And in this case, as you can see, every single one of these commercially available non-Saccharomyces yeast were able to affect the growth of basically the growth of the population of this Hanseospora, meaning there were fewer cells of that spoilage organism when you had this non-Saccharomyces yeast in the mix. And then in that same experiment, they looked at the acetic acid production. So you can see that Hanseospora is producing quite a bit of acetic acid here, and the presence of these non-Saccharomyces yeast significantly reduced that. And again, you can kind of see some are better than others in this case, but most of them are causing a pretty significant reduction in the production of acetic acid. So in this case, the use of these non-Saccharomyces yeast are doing a good job of protecting against sort of the work of this spoilage organism. So before we sort of move on and talk about the experiments that we have on tap for this particular topic, I did want to go back real quick and say, if we look at our list of commercially available non-Saccharomyces yeast, if you look, some of these are specifically for bioprotection, but some of these also have either, in addition to bioprotection, they have some other functions, or they're just listed for other functions that include increasing complexity, increasing aromas and esters and mouthfeel, and giving us some aromatic complexity. So the question is, how are they doing that? And the thing to remember is that these are organisms that aren't just there to protect our fermentation. These organisms sort of have metabolisms in their own right. And they have been shown to have good aroma impact as well as being spoilage organisms. So again, that goes back to choosing the ones that have a positive impact and inhibiting the ones that have a negative impact. So some of the other positive impacts that have been found in various different types of non-Saccharomyces yeast, these yeasts themselves do actually produce some compounds themselves. So there's a number of different types that have been found to be produced by non-Saccharomyces yeast, including esters, which give us some of our fruity and floral characteristics, higher alcohols, which in the right amount can give us some more complexity in our aroma profile, glycerol, which affects mouthfeel, succinic acid, which affects sort of the freshness of the wine, and thiols, which we've talked about quite a lot in the past. But in addition to producing compounds of their own, they also produce enzymes that eventually affect the flavors and aromas of the wine. So they can produce proteases, and those proteases can help just break down the cells to be able to extract more color and aroma and nutrients from the cells. Proteases also break down proteins, and so these could be helpful in helping to make the wine more protein stable by breaking down some proteins. They also produce glycosidases, and what that basically means is these are things that are going to cleave sugar molecules away from other things. And the important thing to remember there is that most of our aroma compounds are not actually perceptible in the juice because they're bound to sugar molecules. They're not volatile at that point, so they don't have aroma or flavor. And it really takes the work of glycosidases cleaving that sugar off in order to unmask those aromas. And so our non-Saccharomyces yeast can play an important role in doing that. They also can produce enzymes that help to break down polysaccharides, which help in clarification and filtration. And then they have a couple of other functions as well. There are some of them that have been shown to actually consume acetic acid, which can reduce volatile acidity. Some have been shown to actually consume glucose and produce products other than alcohol, which help in lower alcohol wines, or when we have really high Brix musts that we don't want to get a really high alcohol wine. And then the other thing is that they can influence the behavior of the microbes around them, including Saccharomyces. And at least in one study, it was shown that having Metschnikowia pulcherrima around actually changed the gene expression of the Saccharomyces in that particular inoculation. And that change in gene expression meant that the Saccharomyces itself produced less acetic acid and more glycerol. So again, in response to having this non-Saccharomyces yeast around, Saccharomyces was affected in its gene expression and its metabolism. It's important to remember when we look at these that there's different genetic types of each microbe. Just like we have different strains of Saccharomyces, there's going to be different strains of Metschnikowia and different strains of Troilospora. And so again, when we look at sort of these commercially available ones for inoculation, different companies have gone through a selective regime on their own and selected their own strain. So Troilospora from one company may not be doing the same thing as Troilospora from another company. They may have been selected for different things, or they just, have different strains of them. And And the other thing there is that if we do want to have some of these positive flavor influences, it has been shown that it is important to give the non-Sacc yeast a little bit of time to work before that Saccharomyces comes in. So when testing whether sequential or co-inoculation made a difference, it was shown that sequential inoculation, meaning non-Sacc yeast, give it a few days, and then adding in your Saccharomyces yeast, did tend to give more impact of these aroma compounds from the non-Saccharomyces yeast. So the first experiment that we have looking at the use of non-Saccharomyces yeast was done by Doug Fabioli at Fabioli Cellars. And this was done in Chamberson. The idea behind this experiment really was that Doug really likes the effect of doing a cold soak and getting a little bit more extraction of color and a little bit more extraction of aroma. But he doesn't really have the setup to do a classic cold soak. So he doesn't have these sort of good jacketed climate controlled tanks that he can then sort of do a red wine fermentation into. He doesn't really have room in the reefer trailer to stick the grapes into. So what he ends up doing is he puts them in, he de-stems into his tea bins and then puts his tea bins in the cellar, which aren't really as cold as we would do for a classic cold soak. And what happens there is that without the cold, it's harder to sort of keep all of the yeast at bay, and the fermentation starts pretty quickly. It's also a time where you have that lag time between harvest and the kickoff of Saccharomyces, where you can get the growth of spoilage organisms. So the idea here was, can we use one of these two methods, the Chitosan at crush or the non-Saccharomyces yeast, to both give us a little bit more time in cold soak and to affect the volatile acidity that might be accumulating during that time. So in this particular experiment, we actually had 3 different treatments. We had a control that just got the normal additions that you would expect during crush. We had one that received Eno-Brett-Org, which is a Lefort product, and it's a Chitosan-based product. And then we had one that used Agide, which is a non-Saccharomyces yeast also sold through Lefort. So just to look at those 2 products a little bit, Eno-Brett-Org, again, is a Chitosan product, and so we talked a lot about Chitosan in part 2 of this video series. In this case, basically this product was developed to be used in wine against Brettanomyces. But when we talked to Sean Richardson at Lefort, he recommended and told him what we wanted to do with this project. This was the product that he recommended for us to try on the must as well. So of the different Chitosan products, this is the one that Sean recommended. In this case, again, this is a Chitosan product. So when we look at the overall cost of it, it looks like it's about $13.50 a barrel, or about 5 cents a bottle. So just keeping the cost in mind when it comes to Chitosan products. The second product that we wanted to look at, or that we decided to use, was Agide, which is it's a non-Saccharomyces yeast product, also sold by Lefort. And this is actually a 50-50 blend of Torulasporia delbrueckii and Metschnikowia pulcherrima. And so we saw examples of both of those in Dr. Osborne's work. And basically, the way that this was explained to me by the company was that they have this mix because they sort of have different strengths. So the Torulaspora that they're using is less sensitive to SO2 than the Metschnikowia, and so it's going to be able to sort of overcome that SO2 at crush a little bit better, whereas the Metschnikowia has slightly better implantation efficiency, and so if it's not hit by SO2, it's going to do a better job of holding space, although both of them basically meet all the criteria that we talked about before. And in their product literature, it basically says with the recommended dose that they give you, within 24 hours, they have 10 to the 5th to 10 to the 6th cells per milliliter. So thinking back to the level of microbial load that we would be thinking about coming in off the vineyard, even on our spoilage grapes, we're up maybe at the max at about 10 to the 6th. And so these numbers indicate that within 24 hours, they're doing a pretty good job of kind of holding that space and outcompeting a lot of the native flora. This one is a lower cost product. It comes in at about $2.13 per barrel. And so if we see comparable results, obviously there is a financial impact of choosing one over the other. So before we jump into the methods and the results of this experiment, I did want to take just a minute to look at Torulaspora and Metschnikowia in particular and look at how they're working. So as we said, both of these strains have the characteristics that we listed in the last slide in terms of SO2 tolerance and or no fermentation and good implantation efficiency. But both of these species have also been shown to produce toxins that affect the organisms around them. Now, how much of those toxins are produced by these particular strains, we don't necessarily know. But I did want to make you aware of how toxins might be working with our non-Saccharomyces yeast for bioprotection. So in Torulaspora, there is a toxin that's been identified. Basically, the paper that I looked at a number of different strains of Torulaspora and tested them against a number of different strains of spoilage yeast. And what they found is that 6 out of the 18 strains of Torulaspora that they looked at seemed to have inhibition of those around them in terms of a toxin, not just holding space, but actually killing those other cells. And when they tested those 6 strains against their starter strain or their Saccharomyces, it wasn't killing their Saccharomyces. So they chose the one that was sort of the most toxic of all of them, and they isolated what that toxin was itself. And that toxin seems to have 2 different functions. It seems to have a beta-glycanase function and a chitinase function. And basically both of those enzymatic functions are attacking the cell walls of other yeast cells. So basically what that means is this Torulaspora is producing an enzyme that's actually eating away the cell walls of the other yeast around it. But for whatever reason, it's not really affecting Saccharomyces. And so some of you might be thinking, wait a minute, chitinase sounds really familiar. Why does that word sound familiar? And if you think back to the beginning of video number 2, we talked about chitinase is something that is also made by, chitinase is a protein that's made by grapes to attack sort of the fungi that might be attacking the grapes. Okay, that same activity is being used here to attack the other fungal cells around it in sort of a chemical warfare between the Torulaspora and the other yeast. And to just give you an idea of what that looks like, that in that paper they basically gave us this figure where they counted the number of different spoilage cells. So we've got some Candida species, some Pichia species, and Brettanomyces, and they basically treated it with this particular toxin or exposed it to this particular Torulaspora strain. And then they looked at what those cells looked like at 3 hours and at 24 hours. So I think Brettanomyces is a nice example for us because that's something that we're pretty familiar with. But after 24 hours, you can see only about 30% of the Brettanomyces cells were still alive at that point. It looks like about 40% were in apoptosis. Apoptosis is a series of, it's a process that the cell goes through as it's dying. So it's this program of different things that happens in the cell during cell death. So it's in the process of dying here. And then it looks like about 30% of those cells are actually already dead. And so this particular strain of Torulaspora does produce a toxin that actually kills a number of the spoilage organisms around it. Now, like I said, I don't know if that's true for the Lefort product or not, but at least we know it's possible with Torulaspora. And not to be outdone, Metschnikowia also produces toxins. So in this particular study, they looked at 7 different strains of Metschnikowia against 114 different yeast strains. And I think the important thing to note here is that so if you look at the figure, these are our Metschnikowia strains, and these are the different strains that they tested them against. And you'll notice there's a lot of zeros right up here. These are Saccharomyces. Okay, and then it's once we get to the non-Saccharomyces species here that we start to see a lot of pluses. Pluses mean it's got some sort of inhibitory effect. And basically they found, and I guess if we zero in here, we can see that, for example, strain number 46 has a lot of pluses. Strain number 48 has a lot of pluses. So that seems, those 2 strains would probably be the ones that we would choose in terms of their toxicity to the other strains around them. And basically what they found is this particular toxin has broad and effective antimicrobial action, including killing other cells without killing the Saccharomyces around it. And so when they dug into this and said, you know, what's going on? How is it that they're killing all of these other strains of yeast? They found that Metschnikowia pulcherrima produces something called pulcherrimic acid that basically once this is produced, it's extruded outside of the cell, and then it binds with iron in the outside environment and forms pulcherrima pigment, which can be seen. It actually has a color to it. And what's happening when that happens is that basically it depletes all of the iron from around from the environment. And it turns out that most of our cells need a little bit of iron in order to act as cofactors in some of their cellular enzymes. And so when there's no iron around, basically it shuts those cells down. And so the way that they showed that this was actually what was happening is they went back and they sort of did the experiment again, but they added extra iron in, and they found that it was no longer inhibitory. So really, the toxic effect here is scavenging all the iron from the environment. And again, in this case, Saccharomyces has another way of getting around that, and therefore it's not affected by this particular toxic effect of the lack of iron. So again, I don't know if the Lefort product actually has this particular toxin or not, but we know that Metschnikowia certainly is capable of this. Okay, so back to the experiment of Fabioli. In this case, again, remembering we have 3 different treatments. So the fruit basically came in the morning, it was processed by midday, and then was in the cellar by early evening. So again, it wasn't cooled down in any particular way, and we're relying on the cellar to try to cool it down. In each case, each one of our 3 T-bins received the same amount of SO2. One of those T-bins just received SO2, one received SO2 plus enobret, which remember is our Chitosan product, and one of those received received SO2 plus agide, which is our non-Saccharomyces yeast product. And again, those were layered in at crush, and much like what you would do with any enzymes, particularly with the non-Saccharomyces yeast, it's important to layer those at a time that's different from your SO2, because even though they're relatively SO2 tolerant, you wouldn't want them to sort of be put in at exactly the same time. So the idea here was to do a 3-day cold soak in the cellar before punching down and inoculating. However, unfortunately, the following day in the morning when they went to do fermentation monitoring, they did see signs of fermentation, meaning there was a little bit of Brix depletion, but there was certainly a cap forming and some CO2 production. And so in that case, because that had already happened, they went ahead and inoculated that afternoon and started fermentation as normal. So at that point, each one of those bins was treated the same. They got punched down twice a day. They got all the same additions. And again, you can look at the report that's posted with this video session to look at all of the specifics of how those were done. And then it was pressed on the same day and inoculated for malolactic, and it went through malolactic in the barrel. So some additional observations for this, again, the folks that were working on this particular project really did make a note to say that this was not perfect fruit when it came in off the vineyard. There was just a little bit of evidence of volatile acidity even on the fruit that was coming in, and that the fruit was pretty warm because, again, it was harvested in the morning, but it was on a very warm day. And so in this case, the fact that fermentation kicked off really fast, in a different environment, we might be able to delay fermentation and get a little bit more cold soak there, but this particular set of circumstances didn't really allow it in this time. So as much as enobret or agide may have been working, it's just not enough to overcome the desire of all of these microbes to start fermentation. Okay, so when we look at the general chemistry here, I took chemistry both in December and in February, and because we're looking at whether or not we have a good antimicrobial effect that's going to affect volatile acidity in the longer term, I thought I would give you the information from both of those times. So I gave you, for each of our bins, we have a December set of samples and a February set of samples. Just to be clear, though, these are from the same barrel, so this is not a situation where we have replicates. This is just basically a time course. So again, we see the general chemistry of these things is not terribly different. Our pHs and our TAs and other things are not terribly different. I will point out, and I should have pointed this out, we had one experiment in video 2 also that had some measurable residual sugar, and we see it here as well. I've seen that a number of times this year as I'm running chemistries, and I didn't see it hardly at all last year, where we had a little bit of residual sugar. Now, these numbers are not anything that you would be able to taste as residual sugar. Most of us don't taste RS until it's maybe about 4 or 5 grams per liter. So this is not making the wine taste sweet. But the reason I point it out is that it is essentially, it gives us an additional risk factor for spoilage. So all of those spoilage microbes that might be still in our juice from coming in off the vineyard, or maybe in the resident in the barrel, or wherever else they might be coming from, this is basically a nutrient source for them. This is a way that they can get energy so that they can metabolize more. And so if you do have any of these residual sugar numbers that are less than or more than 1, it is kind of it's a risk factor to be aware of, and particularly if you're aging in barrels, and if you're aging in barrels for a long period of time. Again, that's just an additional risk factor that might say, oh, maybe I need to take one more step of prevention. Maybe I need to do a preventative Chitosan step, for example. Okay, so to go back then, but this experiment particularly, we wanted to focus on the volatile acidity. So we'll come back to this this column looking at volatile acidity. So it looks like our initial volatile acidity was pretty much the same between each of the 3 treatments. We do have some volatile acidity, and again, this is kind of these numbers tell us that we're pretty close to being able to perceive that VA as well. We're not sort of sub-perceptible anymore. There is some probably, we're close to that. But the other thing I wanted to look at was sort of the accumulation of VA between December and February. Now, normally we would expect a little bit of accumulation. So the question, just due to time and due to barrel aging. So the question is, did any of our treatments sort of help us with that? Did they take down this microbial population in any way that sort of helps us to have a little bit less VA accumulation? And it looks like we may have gotten a little bit of effect from both the aged and the enobret, based on what we're seeing in the control, but it's not a big enough difference to say that that was certainly the case. And if we were to do it again, we would have the same impact. So again, it may be this kind of case where that initial warmth in the grapes just wasn't enough. None of our Chitosan or non-Saccharomyces yeast wasn't going to be enough to be able to overcome that initial drive for these organisms to consume the resources that we gave them in that initial bin of grapes. So we did take color numbers because that's just part of what our normal chemistry gives us, and again, we don't see any differences there, and we wouldn't expect to see any differences. Again, the only reason we would probably expect to see color differences would be, for example, if we had differences in SO2, and in this case, they all got the same SO2. So, and then when we look at microbiology, again, we don't really see big differences, and we certainly don't see the aged or the enobret decreasing the amount of microbes that we see in the control. So in this case, we don't get fewer acetic acid bacteria. And I realize that the enobret looks like it's a 10 to the 6 and the other one's a 10 to the 5, and that looks like a big difference. But if we realize that this is really a difference between an 8.2 and a 10, we're really within the same order of magnitude on all of those. And in this case, it looks like the enobret actually has a little bit higher microbial load. It's my suspicion that that might be reflective of the barrel that it's in versus the wine that we put in the barrel that got the various treatments. So again, in this case, though non-Saccharomyces yeast and Chitosan might be useful tools, in this case, they're not useful to overcome the warmth of the grapes in trying to extend that, that maceration at the very beginning of fermentation. Okay, and then the last experiment that I'm going to talk to you about in this session is one that was done by Daniele at Barboursville. This one was an experiment with Petit Verdot, and in this case, Daniele wanted to see if he could use a bioprotective non-Saccharomyces yeast product to be able to not use sulfur dioxide at crush. So all of the other experiments we've looked at so far, we're using SO2 plus something else. In this case, this is an illustration of that other potential use of these products that could say, you know, can I use this instead of SO2 if I have good, healthy grapes coming in and I want to pull back on my SO2 use for whatever reason. So in this case, we have two treatments of the Petit Verdot. We've got one that received 25 parts per million of SO2 at crush. Again, it was 2019. We didn't really have to add too much SO2. And then one treatment that received no SO2, but instead received Bio Nature non-Saccharomyces yeast, which is a product that is produced by La Mothe Abiette. So just a couple of things about Bio Nature itself. This basically is a strain of Metschnikowia pulcherrima. And in the product literature, basically, they explain that they kind of took 77 different strains of Metschnikowia, and they chose this one. And again, just to underscore the criteria by which we choose these things, this particular strain doesn't ferment, but it does have strong dominance and colonization, so it's good at implanting, but it doesn't ferment. It does have beta glycosidase activity, so in that case, it's meant, or it's expected to potentially have a positive aroma impact in cleaving sugars off and releasing aromas. It has a low production of H2S and volatile acidity, so again, it's not a spoilage organism. It doesn't have those spoilage components to it. It does make the pulcherrimin pigment, which is toxic to many of the microbes around it, many of the non-Saccharomyces products around it. And that includes inhibition or demonstrated inhibition to Brettanomyces, Hanseospora, which again is Cloacra, Candida, but it doesn't inhibit Saccharomyces. And this particular strain is resistant up to about 50 parts of SO2. So again, most of the products that are on the market have similar things, but just to underscore sort of what the characteristics of a good bioprotective yeast might be, you know, that they're all listed here. So, so the methodology here, this Petit Verdot was all hand harvested from the same rows in the vineyard. It was destemmed on the same day, and either received 40 grams per hectoliter of Bio Nature, which is how much they're recommending that you add, or 25 parts per million of SO2, which is what Daniele felt the fruit needed for this particular year. That after destemming, it was must pumped into the fermentation tanks and then held for 2 days for a cold soak, just with a blanket of carbon dioxide and no pumping over. So again, sort of the idea here being, will our bioprotective yeast be enough to control spoilage in that cold soak tank when we haven't added SO2? After 2 days, both tanks were inoculated with FX10 and got the same nutrient regime and the same additive regime and the same punch down regime for the remainder of fermentation. So again, if you want to know the specifics of that, I would recommend that you look at the report that's posted with this video. Both of these received 2 weeks of extended maceration after fermentation, and then they were pressed. The free run, we only used the free run for this particular experiment, and at Barbersville they do malolactic in tank. So after the free run was put in tanks, it was inoculated for malolactic, went through mallow in tanks, and then was transferred to barrels. So when we look at the juice chemistry, this just basically tells us how comparable those grapes were when they came in. In this case, they had the same Brix. Their pHs were a little different and their TAs were a little bit different. That just reminds us that things coming in off the vineyard are not always exactly the same, even when we try to randomize them as much as possible. When we look at the overall wine chemistry, again, we wouldn't really expect to have much difference in our pH and our TA and our alcohol, and we see that they're not that different. There's two things I would like to point out on here, and they're on opposite sides of this table. So if we look at the volatile acidity here, again, remember our question was, can we control the VA production or spoilage organisms by using Bio Nature instead of using SO2? And in this case, we have slightly higher volatile acidity with Bio Nature, but again, that difference of 0.04 is not a big difference at all, and we would really need to do this multiple times to see if that's a real difference or if that's just the difference of, a random difference between the two. But either way, it doesn't seem like we have a significantly large amount of spoilage there, considering that we did have some cold soak in these particular grapes. But the other question was, did this affect the total acidity or the total SO2 at all? Remember, there was none added at crush for for the bio nature, and there was some added to the control. And we do see that there's a small difference in total sulfur, but we don't see a very big difference at all. And that just reminds us that when we're adding that sulfur at crush, a lot of that is getting bound up by our grape skins and is actually being taken out. And so when we sort of do that big add at crush, a lot of times we don't see the results of that in our total sulfurs on the other side. That doesn't mean that you don't think about how much sulfur to add at crush, but it just means that if you're adding 50 parts at crush, that doesn't always mean 50 parts added to your total. Sometimes you don't get all of those 50 parts. So another question that Daniele and I had about this experiment, though, was whether we would see a difference in the color extraction, in the extraction of phenolics, if we didn't use SO2. So one of the things that SO2 does at crush is it interacts with the grape solids to actually help to burst open some of those grape skins, which helps to release some of the anthocyanins. And so sometimes when we have lower amounts of SO2, we see lower amounts of color. So when we look at the color intensity here, we don't really see a big difference in our control versus our bio nature. So it looks like in this particular case, Daniele didn't really need the SO2 in order to get a pretty high color intensity off of this Petit Verdot. So that speaks both to the ripeness of the fruit itself, and also to the fact that that 25 part add wasn't really doing too much to affect the color intensity or the color extraction. We did go ahead and run a rapid phenolic analysis on this as well to see if we would see a difference in the anthocyanins or in any of the tannin component, again, as a result of having used SO2 at crush. And in this case, we can see that the total anthocyanins are a little bit higher in the SO2 than in the bio nature. But when we look at that proportionally, it's not a big difference. And so again, in order to say that that was that was anything other than just that's interesting, we would probably need to do replicates to see if we got that every time or if that was just this particular time around. That may have just been due to random chance. And we don't really see any notable differences in any of the other phenolics. So it seems like in this case there was no price to pay for not using SO2 when it comes to color and phenolics. And then the microbiology, when we look at it, again, our big question here is do we see an increase in spoilage organisms when we're not using SO2? And the answer really is no. We don't see any notable differences in any of the organisms that are listed here. There are some lactobacillus and some pediococcus here that probably need to be mentioned, but they are the same for one versus the other. So the other question here is if this was something we were concerned about, the next question would be, what if we used both? Would we see fewer lactobacillus or fewer pediococcus species on this side of things? So that would be the next question to ask here. But in this case, the first question, can I replace my small amount of SO2 that I'm using in a good vintage with a bioprotective yeast? And it looks like in this case, at least the measures that we looked at in this experiment, that it looks like yes you can, or at least, like I said, this year with this fruit in this experiment, it seemed like that was a reasonable choice. So that was the last experiment that I wanted to present to you today. But I also, before we go, I did want to look at one more thing. When I got these data in, I wanted to look to see how do the microbial data that we saw this year compare to previous years, particularly since this whole set of experiments was born out of a concern from the microbial load that was coming in 2018. So what I did is I went back in the archives of the Winemakers Research Exchange and I found any experiments where we measured the microbial load in the experiment. And I made a big spreadsheet of those, and I've given you kind of the range that I found with those experiments on this table. So just to sort of step through that, in 2016 we had 2 such experiments, in 2017 we had 6 such experiments, in 18 we had 3, and in 19 we had 4. Now I will say that a number of these experiments were for barrel stirring or lees stirring in some respect. And so when it came to those, if there had been any added lees, for example, I just gave you the number that was given for the control. So if we're not adding any lees to the barrel, what kind of microbial numbers do we see? And there were a couple of things that I think that we need to be aware of when we think about that microbial load that's coming in on the grapes and how that's affecting the wine when we get to the other side of the things. So the first thing to notice were that overall the counts were higher in 2018 than in the other years. And we can see that in our acetic acid bacteria, but we can also see that in Pediococcus and Brettanomyces and even Zygosaccharomyces. So when we look at the potential spoilage organisms that are coming in say 2018 versus 2017 or 2019, we do need to think about that at crush in terms of how much SO2 we're using, but then also if we might want to use some of these other tools. So as much as we didn't see a big difference in any of these microbial counts in 2019 with using these additional tools, if we do see another year like 2018, I do think it's useful to keep it in mind that these are potential other tools that we could use to knock back that microbial population in those particular years. And also just to remind everyone that obviously there's no kind of magic bullet that takes all the microbes away. And to be honest, we don't really want to take all of the microbes away. We want to keep the good ones and limit the bad ones. But these are tools that always need to be used in conjunction with wise winemaking decisions and good cellar hygiene. So again, thinking back to that list that we had in session number 1 or video number 1, that there's so many different places that spoilage organisms can take over, and really this is a game of being vigilant at every different step. So the other thing I wanted to say before I close out the video is that in looking through non-Saccharomyces yeast, there are also a number of other non-Saccharomyces yeasts that might be useful to explore if you're interested in this kind of technique in order to fine-tune some of your wines. So for example, there are commercially available non-Saccharomyces yeast that, at least in the laboratory, have been shown to actually consume volatile acidity. That might be a useful tool to use. There are some that can add acidity and reduce alcohol by producing lactic acid. So basically you use these again at the beginning of fermentation, and they're going to take some of your sugar and turn it into lactic acid instead of turning it into ethanol. And that could be a way of acidifying some of these musts and dealing with some high sugar must. And then there are some that consume malic acid. And that particularly would be helpful if we want to think about some additional ways to dealing with the high amounts of acidity that we see in Petit Manseng. So thank you so much for getting all the way through our virtual session of the Winemakers Research Exchange. As I said in video number 1, I really do miss seeing each one of you, and I know that it is not the same when we cannot taste the wines and we cannot discuss them together. I really do miss your comments and your insights and all of the things that you add to this discussion. So once again, I just look forward to the time that we can do this in person again. But until then, if you do have comments or questions or things to add to the discussion, please let me know, and I will try to find a way that we can share those all together. So I hope everyone is staying healthy during this time, and we will talk again soon.